News release, Wednesday, January 31st, 2024. Proteins could serve as biomarkers that improve diagnosis and guide the development of novel therapies. Researchers at the National Institutes of Health detected abnormal proteins in the spinal fluid of people with amyotrophic lateral sclerosis, ALS, and frontotemporal dementia, FTD, which could help improve diagnosis of these diseases. The findings were published in Science Translational Medicine. The proteins in question are built from cryptic exons, abnormal portions of RNA, the cell's instructions for how to build proteins. Cryptic exons occur when TDP43, a protein that regulates how RNA is processed, stops functioning normally. TDP43 dysfunction is linked to ALS, FTD, Alzheimer's disease, and limbic-associated TDP43 encephalopathy, LATE. The study showed that these misspliced sections of RNA can sometimes generate new proteins from the cryptic sequence. The findings advance our understanding of how cryptic exons may be involved in the dementia disease process and could help identify diseases involving TDP43 dysfunction before symptoms appear. Currently, TDP43 aggregates in the brain can only be detected at autopsy. Normally, TDP43 helps control the production of proteins by regulating how RNA is processed. To function properly, TDP43 must remain in the cell nucleus where DNA is located. In ALS and other neurodegenerative diseases, TDP43 leaks from the nucleus and instead forms abnormal clumps in the surrounding cytoplasm. We show that TDP43 pathology causes brain cells to missplice hundreds of RNAs, and that some of these RNAs are translated into new proteins that are not normally found in healthy cells, said Michael Ward, MD, PhD, senior investigator at the National Institute of Neurological Disorders and Stroke, NINDS. Co-director of the iPSC Neurodegenerative Disease Initiative at the National Institute of Health, Intramural Center for Alzheimer's and Related Dementias, CARD, and senior co-author. This conceptual discovery may enable future development of sensitive diagnostic tests to detect TDP43 pathology in living patients. Diagnosing ALS and related conditions can be challenging. There's currently no single test that can definitively diagnose ALS or FTD, and the symptoms can resemble those of other disorders. To help diagnose the disorders, doctors will perform a physical exam, assess the person's symptoms, conduct brain imaging, and use tests to rule out other conditions. TDP43 pathology is a defining feature in almost all patients with ALS and about half of those with FTD. It is also seen in roughly half of patients with Alzheimer's disease. However, current technology can't detect or measure TDP43 pathology in living patients, hindering accurate diagnoses and therapy development. The presence of cryptic exons typically destabilizes and degrades RNA transcripts, the RNA strands produced when a gene is transcribed. Until now, scientists had not explored the possibility that some cryptic exon transcripts are not degraded, but instead generate new proteins. Using induced pluripotent stem cells, iPSCs, that model neurodegenerative disease, the research team developed a catalog of cryptic exons and were able to identify 65 peptides that came from 12 cryptic exons. They then identified those same cryptic exons in postmortem brain samples taken from people who had ALS-FTD spectrum disease and compared to healthy controls. The team followed up by confirming the presence of new resulting proteins in patient-derived iPSCs. They also showed that proteins containing cryptic sequences interacted with other proteins differently, likely altering their function. The authors raised the possibility that these new proteins might be seen as foreign by our immune system, thereby trigger inflammation that could contribute to neurodegeneration. Finally, the team examined cerebrospinal fluid from patients with ALSF-TD spectrum disorders and identified 18 new proteins encoded by cryptic exons in 13 genes. In addition to advancing our understanding of the biologic processes that give rise to neuronal death in ALS, FTD, and other TDP43-associated neurodegenerative diseases, the study's finding of novel proteins in patients' spinal fluid may enable early diagnosis and serve as biomarkers that reflect a therapeutic response in clinical trials. This study was funded in part by the NINDS Intramural Research Program, the National Institute on Aging, NIA, and the National Institute of Health Intramural Center for Alzheimer's and Related Dementias, CARD. This press release describes a basic research finding. Basic research increases our understanding of human behavior and biology, which is foundational to advancing new and better ways to prevent, diagnose, and treat disease. Science is an unpredictable and incremental process. Each research advance builds on past discoveries, often in unexpected ways. Most clinical advances would not be possible without the knowledge of fundamental basic research. NINDS is the nation's leading funder of research on the brain and nervous system. The mission of NINDS is to seek fundamental knowledge about the brain and nervous system and to use that knowledge to reduce the burden of neurological disease. About the National Institute on Aging, NIA leads the U.S. federal government effort to conduct and support research on aging and the health and well-being of older people. Learn more about age-related cognitive change and neurodegenerative diseases via NIA's Alzheimer's and related dementias. Education and Referral, ADEAR Center website. Visit the main NIA website for information about a range of aging topics in English and Spanish, and stay connected. About the National Institutes of Health, National Institute of Health. National Institute of Health, the nation's medical research agency, includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research, and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nih.gov. National Institute of Health, turning discovery into health. Siddiqui S. Misspliced transcripts generate de novo proteins in TDP43-related LSFTD. Science Translational Medicine. 2024, Jan 26. DOI 10.1126/Med 162.